today to Singleron's Innovators Seminar. I'm Andreas Schmidt, the Senior Vice President of Business Development of Singleron, and it's a true pleasure and I'm excited to have with me today Professor Jens Schwamborn, who is a rare species of scientist. He's both a distinguished professor and as well as a successful entrepreneur. He's currently the professor at the Center of Systems Biology at the University of Luxembourg, and before had academic positions at the University of Münster and Vienna. And he's also the founder and CEO of Organotherapeutics, one of the key drivers of innovation in midbrain organoids and the understanding of Parkinson's disease. We are excited to dive today with you into an ocean of knowledge of organoids, vascularization, and neurological models for that. We want to thank you for the opportunity to assist you a little bit on some of the single cell aspects, and we are excited now to listen to your presentation. Today's session is a very popular one with a lot of people listening, so we will leave the microphones closed, but please be welcome actually to share your questions. I will read them out then from the chat. And with that, over to you, Jens. We are excited to listen to you.
All right, thanks a lot for the very kind introduction. I'm not sure whether I can live up to that, but I try to. So let me share my screen and then we can directly dive into that. So ideally, you should see a full screen mode now. And well, I would get started. So I'm going to talk about modeling Parkinson's disease in patient-specific midbrain organoids. So everything that I'm talking about today is about a human brain organoid system specific for the midbrain, and it's a clear focus on Parkinson's. And before I go into actual science, just a disclosure, as Andreas just mentioned, I co-founded Organotherapeutics by now 5 years ago, and many of the technologies that I'm showing you today have been actually developed at the university and licensed to the company. So this paper here is already a couple of years old, but it is still extremely true. We are getting in a situation for Parkinson's disease similar to what we have for Alzheimer's, where we have a real societal issue. If you look at those numbers here, how the amount of patients over the next couple of years is exploding. And if you then think about the cost and the socioeconomic burden that this will have for our society, we have a real issue. And actually, these numbers are already outdated. I guess by now we calculate with about 10 million PD patients. And with the aging societies that we have, this is going to increase dramatically. So we really better get a deeper understanding of the disease and come up with at least disease-modifying therapies. So what's Parkinson's disease? Parkinson's disease is named after the medical doctor who first described it, James Parkinson, and it is mainly diagnosed based on motor symptoms. And I'm sure you all have seen those symptoms in one or the other patient. This is tremor, rigidity, postural instability, and some others. And these are typically summarized as Parkinsonism. However, this is seen as just the tip of the iceberg because the patients also suffer from a variety of non-motor symptoms, and they can be very diverse and at very different timings in the disease course. For example, sleep disturbance and hyposmia can occur very, very early in the course of the disease, often decades before motor symptoms are diagnosed. On the histopathological level, what is happening is that a particular nerve cell type is degenerating. This is a huge oversimplification, so it's a systems disorder. Many regions of the brain and even of the whole body are affected, but for simplicity, we focus particularly on probably the main cell type being affected, namely the dopaminergic neurons of the substantia nigra in the midbrain. So under physiological conditions, these cells have their cell bodies in the substantia nigra, and they project their axons to the striatum where they secrete dopamine. And this continuous dopamine secretion from those neurons is controlling muscle movement. However, during the disease, these dopaminergic neurons degenerate. As a consequence of that, levels of dopamine in the striatum drop down, and we see the typical symptoms. And you can see here two cross sections to a diseased and a healthy brain. You see in the healthy brain these dark staining here, which is the substantia nigra. This black gives it the name nigra, while in the patient, this is largely gone, so we have really massive degeneration. Additionally, like in many other neurodegenerative disorders, we find protein aggregates as a key hallmark. In the case of Parkinson's disease, these aggregates are called Lewy bodies or Lewy neurites. They mainly consist of the protein alpha-synuclein, but also do contain a variety of other proteins, including, for example, tau. So there is positive and negative factors that influence the likelihood to get Parkinson's or not to get it. The major driving factor certainly is aging. The higher somebody is, the higher is the likelihood to suffer from it. Then there's genetic factors. Probably plus minus 10% of all Parkinson's cases are clearly monogenetically driven. And there's environmental risk factors, including, for example, pesticides, but probably also air pollution. On the other hand, there's inverse risk factors. First is female gender, probably having to do with certain hormone levels. Then there's obviously lifestyle factors, like if you live a healthy life, doing sports, eating healthy, this is obviously good. But there's also two unexpected things. And this is caffeine intake and smoking. Both seem to be protective. And then there's genetic factors that can be protective. So if we look at the time course of the disease, and we look at here just first on the right side of the diagram, if we define time point 0 as the time point when the disease is diagnosed, we see that at that stage already plus minus 50% of the dopaminergic neurons are lost. We have a start of the motor symptoms. This is this dark blue triangle here. But we also see that non-motor symptoms are already there for probably 10 or even 20 years. So as I say, these non-motor symptoms really precede the disease by decades. And we have here this progressive loss of the dopaminergic neurons shown in red, also preceding disease onset by decades. And only when half of the cells are gone, the disease really is diagnosed. And what we are wondering in many of our projects is whether there's actually a phase even before these non-motor symptoms, actually even during development, that predisposes for degeneration at later stages. So that brings us to something what we call a multiple hit hypothesis for PD. What we speculate here is that there is a neurodevelopmental component. So something during brain development is not happening as under physiological condition. And this could be, for example, driven by mutations in one or many other Parkinson's associated genes. It not have to be genetic. It could be also, for example, a viral infection, but a genetic is obviously easier to understand. And that will be like the first hit. If that is followed by a second hit at a later stage in life, which can be simply aging, it can be brain injury, it can be environmental factors, then it really comes to massive neuronal cell death. First non-motor symptoms would occur, followed by increasing loss of the dopaminergic neurons, more than 50% being gone. And then the disease is diagnosed on the motor symptoms and takes its well-described course. In order to study this process, we make use of a human midbrain organoid system, and that is really where I'm going to focus in the presentation today. So we start with a sample from a healthy individual or a Parkinson patient. The sample can be a skin biopsy, blood, can be different cell types. These are then reprogrammed into induced pluripotent stem cells. And from the induced pluripotent stem cells, we do not directly differentiate into the midbrain organoids, but we have a stable intermediate, which are human neuroepithelial stem cells. And we do that because these neuroepithelial stem cells during the derivation process are already pre-patterned for the midbrain hindbrain. So we quite strongly restrict their differentiation potential, which also strongly restricts the variability of the human midbrain organoids. In other words, that makes the whole protocol way more reproducible. Just in a nutshell, what are midbrain organoids? These are three-dimensional structure up to 4 millimeter in size. They have midbrain identity, expressing, for example, transcription factors like FOXA2 and LMX1. They have lots of dopaminergic neurons that we can see here with the TH staining, but also other neuronal subtypes. I come to that in a second. These dopaminergic neurons produce dopamine. We can see that in a staining here in green, but also secreted in the medium. Interestingly, midbrain organoids form neuromelanin, and neuromelanin is a factor that we only see in the brain at later stages of development. So this is nothing that we would see in the embryo, and this is also nothing that we would see actually in mice and rat. So it's really a mature feature, and that's important for us because maturity of these organoids is obviously important in order to model Parkinson's. The organoids show self-organization, so it's not just a clump of cells. We really have a tissue layering, and as a true organoid, it's obviously not only one cell type, not only neurons, but we have also glial cells, including astrocytes and oligodendrocytes. The neurons in the organoid show electrophysiological activity, and a subset of the dopaminergic neurons even show pacemaker firing, as we would see it in the healthy human midbrain. They fire with a regular frequency of about 6 Hertz. So first thing that we wanted to know, what is actually in there? And to really understand what are the cell types that occur in midbrain organoids, we started here with a single-cell RNA sequencing approach that was done together with colleagues in Luxembourg, Alex Kupin's lab, as well as in Japan, in Yokohama at the Riken Institute. And you see here one of these typical single-cell RNA sequencing plots. Every dot is a cell, and every color is an identity. And if you look at 2 time points, what you can already see is that from day 35 to day 70 of differentiation, complexity increases. So we have more different cell types specified. We find the expected suspects like dopaminergic neurons, astrocytes, but we also found other neuronal cell types like glutamatergic neurons, GABAergic neurons, and serotonergic neurons. So we really recapitulate the variety of neural complexities that we would see in the human midbrain. And we also find more exotic cells like these ectoderm-derived COL1A1 positive cell type that also recently actually has been described in the human brain. However, we obviously here have very clear limitations because we derive these midbrain organoids from the neuroepithelial stem cells that I've mentioned before. They contain only cells that come from the neuroectoderm, but everything that comes from the mesoderm is not there. By definition, we start with committed cells. So what we are missing is 2 very important structures. These are microglia as well as blood vessels. I'm not going to talk about blood vessels today. We have work ongoing on that and maybe in the next webinar, but not for today. But I will focus on the microglia. So the microglia are the immune cells of the brain. They have, particularly for Parkinson's disease, a huge importance because neuroinflammation is contributing strongly to Parkinson's. So we're really eager to get microglia in our midbrain organoids. Just going a little deeper into the functionality, microglia have a variety of functions. Everything around the immune function is, antigen presentation, production of chemokines and cytokines. They are very active in phagocytosis. They can undergo aging and senescence. This is a local population of the brain, so they can divide and produce, basically maintain their own population. Very interesting is that they conduct synaptic pruning, so they are able to remove synapses that are inactive, and there is functions in trophic support as well as contributions to the blood-brain barrier. So a huge variety of functions, and that will be certainly a very nice enrichment for the brain organoid system. There was a report from Sally Cowley's lab at Oxford describing to derive macrophage precursor cells from iPS and then further differentiating them in microglia-like cells. Microglia at the end of the day are probably nothing else than the macrophages of the brain. So we teamed up with Sally's lab. A student from my lab spent some time there, learned the method, and we transferred it here to Luxembourg. And quite nicely being able to produce these microglia in the dish. Here you just see a 2D culture. So we can take iPS, differentiate them into microglia. They express IBA1, PU1, typical markers, these P2RY12. And in Western blot, you see other markers. In a PCR approach, you can see your other markers that are robustly expressed. And we're quite convinced that these cells have microglia identity. So the challenge was now taking these microglia from the 2D culture and bringing them into midbrain organoids. But before I come to that, just a slide about the functionality. So as true macrophages, you obviously should undergo phagocytosis. So we're treating them with these zymosan particles, and they are shown here in red, while in green you have an IBA1 staining showing microglia identity. And what you can see is that the microglia indeed take up these particles. So you have here 2 3D reconstructions of the images, really showing that the microglia end up in the cells, but you can also see that in the video here. So the particles are shown here in green and not in red, and the microglia are in bright field, and then you can see, particularly in the higher magnifications, that they're indeed very active in phagocyting these particles, so they are functional. So what we now wanted to do is get our microglia into the midbrain organoids. And what sounds quite trivial actually took us probably about 2 years, because we had to find media conditions that on the one hand are good for the microglia, so keeping them alive and happy, but on the other hand, also keeping the organoids alive, in particular the dopaminergic neurons in a good state. Because we typically have the issue that whatever the microglia liked was killing the dopaminergic neurons and vice versa. So we had to come up with a strategy which eventually worked, and we now have conditions that allow this. So what's now possible is to take an IPS line and on the one hand derive the midbrain organoids in the way I've shown that to you already, but then you can take the same IPS line, derive these macrophage precursors, mix them in the midbrain organoid, and they will migrate in there and be active. And you can also build mosaics, obviously. So you could take one IPS line, produce organoids, and then take another IPS line and produce macrophage precursors and then microglia, and thereby understand whether, for example, disease processes are more driven by the organoid or more driven by the microglia. And we actually did that quite intensively, building these mosaics. Actually, under physiological conditions, it seemed not to matter whether it's the same IPS line or different lines. Under disease conditions, that's very different. So here's just some images and also a representation how the process works. So we start with seeding of the organoids. We would produce them until probably 15 days of differentiation, then mix in the microglia and do at least 20 more days of co-culture, but you can also go for longer time points if you like. And what you would then see in a midbrain organoid here in the abbreviation MO is that you have midbrain identity, like you have FOXA2 and you would have TH staining. But then in the assembloids, so assembloid would be the term I'm now using for the organoids plus the microglia, you would have the midbrain markers still, like FOXA2 and TH, but on top you would have IBA1 as a marker for microglia. And in the protocol that we are currently using, about 6% of all cells of the organoid would be microglia of the assembloid. And this is pretty close to what we would actually see in the physiological human midbrain. Now, to further understand what is the nature of those microglia in the organoids, but also what are these microglia doing, particular to the neurons of the organoid, we used a single nuclear RNA sequencing approach, which was actually done together with Singleron. And we here compare the midbrain organoid versus one of the assembloids. And on the first glance, you see, well, the pattern look pretty similar, but then you can see this little population here in pink labeled MGL. This is a microglia that we would only see in the assembloid and not in the organoid. And also then in a hierarchical clustering, they obviously totally cluster away from all other cell types, which makes lots of sense because essentially they represent a different lineage. They come from the mesoderm, everything else is ectoderm. Also interesting to see here in this clustering is that we can even distinguish the A9 and the E10, A9 and E10 dopaminergic neurons of the midbrain organoid. In Parkinson, only the A9 is heavily affected, while the A10 is pretty unaffected. So let's have a closer look at those microglia. First of all, we see a core signature of microglia genes being highly expressed in them. I already mentioned IBA1 and PU1, but you see here also a bunch of other genes that are all very high expressed, so indicating these microglia in the organoids really have the right identity. And also a bunch of genes down here that are important for phagocytotic activity are highly expressed, again, supporting their functionality. And we do see an array of chemokines and cytokines that are only expressed here in red in the microglia, but not in any of the other populations. There's two IL-6 and IL-7 that are actually expressed by neuroblasts or the neurons, but everything else is strongly microglia driven. And this is not only true on the RNA level, but we can also measure the cytokine release in the medium. You have here on the left side of this clustering, the midbrain organoids where levels are pretty low, and then you have three different microglia lines in organoids, so assembloids, where these cytokine and chemokine level are very high. So we get functional microglia in those midbrain organoids, forming an immune-competent assembloid. Question is, what is that now doing to the actual neurons? And we here focused on the synaptic genes because, as I told you before, microglia are involved in synaptic pruning. So we speculated if there is microglia, we probably have more pruning. So the expression of genes that are involved in microglia functionality, in neuronal synapse functionality should be affected. And there's a lot of diagram on that slide, probably focus on the purple A9 dopaminergic neurons, because these are any of the most interesting for us. And you can see that in pretty much all cases, these synaptic genes like synaptophysin or DCC are downregulated in the blue case, which is the assembloid, in comparison to the organoid. So when there's microglia, these genes are downregulated. You can also see the same thing on Western blot, in this case for VAMP2. So these microglia seem to have a clear effect on the signature, on the synaptic signature of the dopaminergic neurons. We can also see the same thing if we look for a bunch of different genes that are more involved in synapse functionality. Here only focused on the A9 dopaminergic neurons. Pretty much all of them are actually regulated, and here it's also the same in the A10 dopaminergic neurons. So there's good indication that when there's synapses, when there's microglia, synapse function and synaptic gene expression is affected. And if that's the case, we would expect that it has an impact on the electrophysiological activity or properties of those neurons. So we did some patch clamping in the midbrain organoids, or better assembloids, that was done together in collaboration with the University of Heidelberg. And indeed, what we see is that the action potential threshold of those neurons is significantly reduced. So it is easier to evoke an action potential in a neuron that sits in an assembloid, so in the presence of microglia, versus a neuron that sits in an organoid, so in the absence of microglia, indicating that they are probably more mature, which we would have expected. And this is supported by multi-electrode array experiments, where we took midbrain assembloids, put them on the electrodes, and then measured different features, including the interspike interval, so the time between the different firings. And we can see that this is reduced in the assembloids in comparison to the organoids, again, indicating higher degree of maturity. So question is now, having this model in hand, can the organoids be used to actually model Parkinson's disease? Because that's why we all do all that stuff. So we really had to develop quite a bunch of approaches and methods, because these 3D cultures in many aspects are different when you analyze them in comparison to classical 2D neuronal cultures. And a very classical system to study or to induce Parkinson's, particularly in mouse models, is treatment with a toxin called 6-hydroxydopamine. This toxin is taken up by the dopamine transporter. As I said, it's taken up by the cells, it's metabolized, it produces reactive oxygen species, and then at the end of the day, induces cell death. So we did these experiments in our midbrain organoids and then developed a whole pipeline that we highly automated to analyze the organoids after treatment. It goes via sample preparation and then the image acquisition with a high content image analysis approach, and then a few, actually more than a few, image recognition algorithms, and also a machine learning based analysis of the wells of data that comes out there. And if we just very simply look at it, we have here an image of dopaminergic neurons in the midbrain organoids indicated by a TH staining. And then these image recognition algorithms take up the image, draws a mask around each neuron, and then allows us to extract a bunch of different features. Like, for example, from this mask, you could count, obviously, how many dopaminergic neurons you have, how many neurons do they have, how long are the neurons, are they fragmented? So all these kind of features we can see here. And if we compare a healthy control situation with 6-hydroxydopamine treated, we see we have less dopaminergic neurons after toxin treatment. So in a way, we model here an aspect of Parkinson's, and we have a higher degree of neurite fragmentation, which is an indication of degeneration. That works, but 6-hydroxydopamine treatment is really very artificial, right? You put it on the organoids, 2 days later, you see strong phenotypes. This is not how Parkinson works. So that's why, we published this approach, and we particularly published also the algorithm and the computation approach to make sense out of it. But coming now to the key characteristics of PD that we particularly analyzed in genetic forms of Parkinson's. There's probably 3 main features. One is the loss of dopaminergic neurons. The other one is the aggregation of alpha-synuclein. And the third one is the occurrence of neuroinflammation. So I guide you now through a handful of experiments concerning these characteristics. Here is a study where we took IPS from healthy individuals as well as from patients with a LAC2 G2019S mutation, and in 2D as well as in midbrain organoids, we differentiate them. And we really start with the same IPS line and do everything in parallel. If you look in the 2D situation, in the control and the Parkinson patient, we have pretty similar amounts of dopaminergic neurons. So we do not see this loss of dopaminergic neurons as you will see it in the patient. These neurons here in the 2D culture still would show phenotypes. Like I would, for example, be able to see that their mitochondrial functionality is affected. But this pronounced loss of cells is not happening. But if we do the same thing in a midbrain organoid, we very, very clearly can recapitulate the loss of dopaminergic neurons, as you can see here in the diagram. So that was a really striking argument for us that it makes sense to continue with the organoids and focus on them. A second feature that's very important is protein aggregation. And what you see here on the one side is midbrain organoids from a healthy individual. We have alpha-synuclein, it's a synaptic protein, so we would expect it to be there, but we do not have the phosphorylated version of alpha-synuclein, the P129. But if we do the same midbrain organoids from Parkinson patients, we see immediately alpha-synuclein is much higher, but particularly this phosphorylated version comes up, and in a zoom you can very clearly see these aggregated structures that are positive for phosphor 129 in alpha-synuclein. And it's important to mention that this is completely endogenous. This is not triggered by toxin treatment, fiber treatment, or anything. This is really an endogenous development. And if you compare that to what we would see, for example, in the post-mortem brain sample, it looks pretty similar. Like you have here on the right side, synuclein pathology in a postmortem human tissue. You see these Lewy bodies as well as the Lewy neuroids, and we see very, very similar structures like filamentous ring and dance-like structures also in the organoids. So again, a good argument for us that this might be a very nice system to go into the details of Parkinson's. And then the last feature here that I would briefly want to touch on is neuroinflammation. As I said, Parkinson goes hand in hand with inflammation. Whether it's cause or consequence is debated, and it's probably different for different cases of PD. But anyhow, we do see elevated levels of pro-inflammatory cytokines, like here interleukin-1 beta in the synuclein triplication or TNF-alpha in the LAC2 situation. So all these features are nicely recapitulated in the midbrain organoids. And I just want to show you one more example where we derived midbrain organoids from patients with a GBA mutation. So this is a study based on 3 control individuals as well as 3 Parkinson patients with a GBA mutation. Here it's the N370S mutation. Just has been published probably a year ago. And I just want to show you one result which is quite striking. When you look at 3 healthy control midbrain organoids, you can see a very robust differentiation and staining for TH here in red. But then if you go for the 3 patients, you can very clearly see, really on the first glance, you don't need lots of quantification. The differentiation or the survival of those dopaminergic neurons is highly affected. In PD, we have way less of these dopaminergic neurons, and we saw that at various time points that we have investigated. Parkinson, as I said, is complex. It's not only dopaminergic neurons, also other cell types are affected. And in this study that I'm just briefly going to show you here, we looked actually at astrocytes. So this is a very unique situation that we had the lucky possibility to team up with Nobutaka Hattori's lab at Juntendo University. They had post-mortem brain samples from 3 individuals that had mutations in the Parkin gene. And what they saw in histopathology as well as in Western blots is that markers for astrocytes like GFAP are strongly downregulated, particularly in the individuals with a Parkin mutation. Maybe to some extent also in what's labeled here PD, which is idiopathic PD, but for Parkin it was very obvious. And from 2 of these 3 individuals, they also had IPS. So they gave us those IPS, we produced midbrain organoids, and then stained for astrocytes. And the result is pretty striking. Like in the dish, in the organoids, we can very nicely recapitulate the pathology that we see post-mortem. In the healthy individual, we have robust GFAP staining, very robust differentiation into astrocytes. while in the patient, astrocyte differentiation, as you also see here in the quantification, is highly affected. So we see exactly the same thing as we have seen in those post-mortem samples. We can not only recapitulate dopaminergic neuron pathology, protein aggregation, neuroinflammation, but also astrocyte-related phenotypes. And importantly, we can not only recapitulate pathology, but we can also use that to test potential therapeutics. I just show you here 2 studies that have been published in the last years. This is a study where we used midbrain organoids from patients with a mutation in PINK1. And here we tested a compound that actually activates the lysosome, so we get more autophagy. And by using these compounds, this is cyclodextrin, 2-hydroxypropyl cyclodextrin, we can turn on autophagy, get more lysosomes, and actually rescue loss of dopaminergic neuron, at least to some extent, as you can see here in the quantification. And this is in the second study where midbrain organoids with a mutation in DJ1 have been produced. And we knew that this mutation is actually a splice site mutation. So we could treat with compounds that would mediate a read-through at the splice site, and actually thereby would restore production of the RNA and the protein. And this is quite nicely restoring also the midbrain dopaminergic neurons. So that can be used to see pathology, but also to rescue pathology in the dish. And we really hope that in the coming years, this will be heavily used to develop new therapeutic approaches. But again, Parkinson is complex. It's not only different cell types, it's also different brain regions. So we also wanted to mimic this, and we do that by going for another level of assembloids, namely by putting together different brain structures. I told you in the introduction, under healthy conditions or under physiological conditions, dopaminergic neurons in the midbrain project to the striatum to secrete dopamine. While in our midbrain organoids, this is obviously not possible. These neurons grew out, but there is no target. So we gave them a target. We produced striatum organoids and then fused midbrain organoids to striatum organoids to really recapitulate the nigrostriatal axis. And I just show you some images about the assembloids showing their identity. In this case here, for example, we used midbrain organoids derived from iPS that were positive for GFP. So we could always see the green cells come from the midbrain. And you see these midbrain cells express FOXA2, so they really have midbrain identity. While we also see that they, for example, express curine. And we do see that the other sites, the striatal organoids, would express markers like CTIP2, or also what you can see down here, DARP32. So we have these 2 organoids fused to each other, forming an assembloid. And obviously now we want to see, are they really communicating with each other, and what is their identity? Again, to going for identity, we used a single nuclear RNA sequencing approach. We get this kind of a map here indicating the different cell types. You can again see the A9 as well as the A10 dopaminergic neurons, but you can also see the medium spiny neurons here that we would find in the striatum. We have different distributions of those cell types. So the assembloids are quite nicely recapitulating the identities that we see in the physiological human brain. To look whether there's really a connection, we used several approaches, and I show you a few. One is obviously just microscopy, just looking whether these dopaminergic neurons form the midbrain organoid project to the striatum. And we can here, for example, follow an individual cell and then reconstruct the axon. And you can quite nicely see that these midbrain organoids sitting here at the edge of the assembloid is projecting its axon deeply into the striatum. And by using sensors that we can stick either in the striatum or in the assembloid, we can measure levels of dopamine. And in an isolated striatum, you would see hardly any dopamine. But if you put a striatum into an assembloid, so connected to a midbrain organoid, levels of dopamine in the striatum increase dramatically, indicating that the dopaminergic neurons from the midbrain secrete their dopamine in the striatum. And we corrected that obviously for dopamine that probably comes just via the medium route. So this is really a direct connection and direct secretion. Another way to look at connectivity is a rabies virus tracing, and that was done together with the Pasteur Institute in Athens. Here we infect the assembloids with a rabies virus that can only spread via synaptic connection. And actually, you infect the striatal organoid, and with GFP as well as an RFP expression version or compounds of the virus. And when you can detect RFP positive cells in the midbrain organoid, this is a proof that there is a synaptic connection between striatum and midbrain, because it's a retrograde tracing. And indeed, as you can see here in the midbrain side of these assembloids, you can see these red dots that are indicated to be neurons. So they are proven to be in direct synaptic connection to the striatum. And if we go into further details, we can see that at least some of those cells have dopaminergic identity. So they are positive for the red fluorescence as well as for the TH. So we're pretty confident that there is a connection. And then to really finally prove that there is functional connections, so that there are action potentials traveling from one side to the other, we used microtunnel devices that are built by the Lutke lab at the Technical University in Eindhoven. So we have here two compartments. In one compartment, we put the midbrain organoid. In the other compartment, we put the striatum organoid. They are connected by these tunnels. And at the bottom of the tunnels, there's electrodes spotted. And then we can see the firing or the activity of these electrodes when there's an action potential traveling along axons that span through them. And when we did that first, we got lots of signals, but we were computational, not really able to make sense out of that. But luckily, then we were able to team up with Mario Bortolozzi's lab at the University of Padua and at the BIM in Padua. And they really were able to make nicely sense out of those data. And what they were able to see and to show is that there's a very, very clear directionality. So nearly exclusively, these signals are traveling from the midbrain to the striatum, pretty much as we would see it under physiological conditions. So it really rebuilds the nigrostriatal pathway. Finally, what is missing in this whole system is aging. I told you initially that aging is a key risk factor for Parkinson's disease, and our organoids and assembloids derived from stem cells by definition are pretty young, so they are really not aged. But a way to induce artificial aging is by overexpression of progranine. There's quite a bit of work in the literature that really nicely shows that progranine expression can induce aging, can have a strong effect on the nuclear lamina and induce phenotypes that in many aspects resembles aging. The Edenhofer lab at University of Innsbruck is really specialized in this technology, so we teamed up with them. We got some IPS that do have progranine in their genome in an inducible manner, so we can treat them with doxycycline, which on the one hand triggers expression of GFP, but most importantly triggers expression of protein. So these cells that you would see here labeled in green should be artificially aged. And indeed, we do see aging markers in there. I don't go into all the details of all the markers. I just show you here one example, which is the H2AX. And you can nicely see that when you treat with doxycycline, so aging is induced, we see that this marker goes up at day 30, but even more pronounced at day 60. And we can quite clearly see that, these induced aging processes happening. We even see signs of senescence. This is beta-gal staining, and you see that under normal conditions in either organoids that do not have this genetic engineering or that are not treated with doxycycline, we do not see this blue staining, which indicates senescence, while in midbrain organoids that do have the genetic engineering as well as have been treated with doxycycline, this senescence marker comes up, strongly indicating aging. And if you do a PCA of gene expression data, you can also very clearly see that the aged midbrain organoids cluster far away from all the other groups. So indicating aging induction is possible. And with that, I want to conclude it here so that we have enough time for questions. I've shown you that indeed midbrain organoids recapitulate many aspects of the human midbrain. We can use them to recapitulate Parkinson's disease. And we can go even for more complex models like midbrain striatum assembloids. You can induce aging in them, and they will model early stages of Parkinson's disease. All this work has been done by many collaboration partners that you see on the one side here. I try to highlight some of them at least during the presentation. And in particular, I would like to thank my team here at the Luxembourg Center for Systems Biomedicine. I'm not highlighting anybody particular because I've shown you so many different projects where different people have contributed, but trust me, they are all amazing. And I would like to thank, obviously, the funding agency here for the agencies for supporting our work. Thanks for your attention. I'm happy to take questions.
Well, thanks a lot for a fascinating journey through a really, really complex disease, but also actually guiding us through like some very sophisticated methods and technologies to unravel a deeper level of understanding there. Maybe let me start with one question and then I open the floor. So what is the one question that was not, what is the one experiment that you could think of that was not possible so far to conduct that you would love to conduct next?
So the working hypothesis is that Parkinson's disease has a developmental contribution. And we can model that in the dish, obviously, but we have the issue that we cannot really prove it in a human, right? Because we will not be able to look at signs or early symptoms of PD in for example, a newborn, because we obviously don't know whether somebody 60 years later develops Parkinson's. And there is now some interesting work on Huntington's disease, where they had the situation that in aborted embryos that had the mutation for Huntington's, they could very clearly see the neurodevelopmental component. That's an experiment that I would really love to do for PD.
So let me read out the first question from the audience. Do you think introducing vasculature will change the model's performance, especially for drug treatment and its penetration into the assembloid?
Yeah, so we are close to send out the publication for the vasculature, so we have that more or less ready. It has an impact on the performance. It certainly increases maturation. It certainly also decreases hypoxia, particularly in the center of the organoid. For the drug treatment, that's a very good question because the blood-brain barrier is obviously one of the key problems for CNS drug development. The vasculature, as it is now, goes obviously in the organoid, where the organoid are embedded in the medium, and it would typically add the drug in the medium, so it would not have to cross the blood-brain barrier. So for that, that will be, at this stage, not the right model, unfortunately.
Okay. So another question. Would you recommend single cell or single nucleus sequencing for midbrain organoids, especially if multiple cell types are interested? I mean, obviously, I'm interested in that as well, of course. Like we have a history of going through that question actually quite a bit.
Yeah. But yeah, we did both. And so far, my feeling or my observation is that the single nuclear is probably superior because we have less bias in which cell types are enriched. Both methods will lead to a bias in the cell types that is more represented. But for the single cell, we're probably losing quite a bit of the neuronal transcriptome. And that's not so much the case for the single nuclear.
Yeah, sure. So then there's a question from Amina Kama. What is the maximum degree of maturity you have achieved? What do you think about organ on a chip? Is it relevant for your study on PD?
I would not know how to quantify maturity. So the maximum duration that we have achieved is a bit more than 400 days. So it's possible to keep them in. We probably could have kept them longer, but at some point you want to look at your experiment. I would say that for neuronal functionality, based on things that we have looked at, probably would not see massive increase after 100 days. But I also have to say that we would not have investigated that really enough to make there a solid statement. Then organ on a chip, we do quite a bit of on-chip approaches. In many aspects, I'm probably not 100% convinced that it really adds. However, I've shown you this one experiment with nanotunnels or microtunnels. There it was extremely helpful, particularly because it allowed us to do much better multi-electrode array experiments because all the axons were really confined in a very small space on the electrodes. We gave a very nice signal-to-noise ratio. I think the on-chip approach has some interesting promises, like if you can induce sensors, it would be very nice having live readouts. If you can increase the scale, so I have just higher amounts. So I think it has interesting perspectives, but it's probably not there yet.
Yeah. I mean, please do actually post other questions as well. But let me ask one. At the very beginning, one of the first slides, you showed this hit one, hit two model. What do you think do we understand better? The hit one so far, the genetic composition and like the very early onset, or like the environmental factors and the other topics that lead to the hit two?
Certainly the hit two, because for the development, we certainly don't have as many data as for the things that happen later in life. That certain pesticides induce Parkinson's diseases, this is crystal clear. There's also that aging is a major risk factor as a second hit. This is also very clear. So this is much better understood than the developmental contribution.
Are there other questions in the audience about Parkinson, about any of the methods, technologies used? I mean, I'm curious to hear from you, like what of your models could be probably transferred as technology, but also as insights to other neurodegenerative diseases?
So particularly having the midbrain organoids in hand, what we want to do in the future is using them for something that I would probably in many aspects look at the opposite of Parkinson's, which probably could be schizophrenia. Like in schizophrenia, the A10 dopaminergic neurons are more affected, at least in the dopamine hypothesis of schizophrenia, and they actually produce probably too much dopamine. So it could be very interesting to model that in the dish.
Interesting. Are there any other questions from the audience? I know we actually have a large crowd of people listening. Yes, there's one more. Two more. What are some possible reasons that the 3D model better recapitulates Parkinson's pathology while 2D culture using the same cell sources failed to do so?
Yeah, that's a good question. There's probably not a single reason. On the one hand, the 3D cultures are more complex in their cellular composition, so we find simply more cell types. We are thereby more closer to the physiological situation. We have obviously more cell-cell interactions in 3D. I think particularly the presence of other cell types like astrocytes plays a huge role in the pathology. And then there's probably a very practical reason. If you think about a 2D cell culture, like these neurons sit on the plastic and then they are surrounded by medium, which is full of sugar, antioxidants, so all the good stuff. They are really under excellent conditions. While in the 3D, this is more restricted. Which probably by definition makes them a bit more vulnerable.
And then, now there are several more questions tripling in. So I'm curious about the similarities between midbrain organoids and the human substantia nigra in terms of their transcriptomic profiles. How closely do the gene expression patterns in organoids resemble those of mature dopaminergic neurons in the substantia nigra? And what are the main differences you've observed?
Yeah, so we have done this comparison to the adult human midbrain dopaminergic neurons or tissue in one of the papers. So the identity is about 70%, which is probably okay for an in vitro model. And the major difference is very clearly the degree of maturity. We find lots and lots of progenitors still in the midbrain organoids that we would not see in a human midbrain. Because this is completely post-mortem. And then in our organoids, when we did that, everything that is not coming from the ectoderm is obviously not there.
Then there is another question from Amina Kama. What do you think about the gut-brain axis?
Yeah, very interesting. We did quite a bit of work in trying to model that, at least to some extent. I haven't shown that today. So what we want to do there is like building recapitulating the axis, like taking midbrain organoid, hindbrain organoid, enteric nervous system, and intestinal. So we have the components we do have. We also have some of the fusions, but we don't have the full thing. So I think it would be very exciting to model that in the dish. And my understanding from the literature is that we probably have two types of Parkinson's, which is the brain first, where we would see pathology in the brain, and then it probably would spread to the gut, as well as the body first, where we would see pathology first, for example, in the intestine, and then via the vagus nerve, it would be transported up to the brain. Probably both forms exist.
Hmm. And then there's a question from Kyprula Christodopoulou. Would you use the MO model for the drug repurposing research?
Yeah, sure, certainly. We use the midbrain organoids, particularly within organic therapeutics, for drug discovery. It probably doesn't matter when we're talking about repurposing or identifying of new compounds, the assays would be the same. Definitely, I think that this is a good model for doing that. Otherwise, we wouldn't have started the company on it. The hope is probably particularly that we would reduce failure at later clinical stages, because already early on we would test in a human patient-specific model.
Let me ask you a question from the perspective of Singleron as a company. So I'm sure actually you're keen on a keen observer of like all the different fields where organoids are used. Like what fields do you feel, what indications actually is there most excitement, most advancement using organoids right now? I mean, excluding obviously Parkinson and midbrain organoids.
Obviously. I think probably the most low-hanging fruit in a way is everything that is developmental disorders, because that's probably where the model naturally is tuned for. I think it starts to tell us a lot about brain development, or generally organ development. I'm obviously a bit biased towards the brain, but generally being able to recapitulate in the dish what happens during embryo development is exciting. And I think it will be very informative in the years to come to see particular differences between human and model organisms. Yeah, that's probably it.
Are there any last questions? Watching the time a bit, I think we have a minute left maybe. If there's a burning last question, then shoot now. I mean, otherwise, I would like to thank you actually for like a really fascinating talk.
Thank you.
Like spanning the whole arch from like understanding a complex disease to the various technological ways actually how to dive deeper into that. And I have to say it has been a pleasure, not just in this conversation today, but also actually working with you on multiple of these aspects. So thanks a lot. And well, if you enjoyed this seminar today, please also register for the upcoming webinars. And thanks a lot to the lively audience, many questions. And well, we are looking forward to invite you again to many of the extension of the questions and aspects of what you were presenting. Thank you.
Pleasure. Thank you for hosting. Bye-bye.